Welcome, everybody. It's a real pleasure to launch this first scientific update on highly pathogenic avian influenza virus. Many thanks to CIHR and a planning and dissemination grant. So my name is Samira Mubarak. I'm one of the infectious diseases physician and medical microbiologist at Sunnybrook Health Sciences Centre. I'm also a virologist at the University of Toronto. I have the honor and privilege of living, working, and collaborating on the treaty territories of the Mississaugas of the Credit First Nation, as well as the traditional lands of the Anishinaabe, the Haudenosaunee, the Wendat, Huron-Wendat, Chippewa, and Ojibwe First Nations. So in terms of what we'll be talking about today, we're really going to be focusing on the biological aspects of highly pathogenic avian influenza virus. And the learning objectives really are quite broad. We're hoping to enhance current knowledge on interactions between highly pathogenic avian influenza virus and host factors, and also touch on its pathogenesis. We'll be advancing our understanding of HPAI transmission risk and implications for both human and animal health. And then we'll close out the discussion to talk about countermeasures, including vaccines. So before we move on, I want to give many, many thanks to CIHR again for supporting this seminar and knowledge dissemination event. And very special thanks to Aaron Love and Andrew Matejic from the Canadian Association for HIV Research, who are supporting this from an operational perspective and logistical perspective. So many thanks to both of you and to CAR and also to CIHR. Also, many thanks to my co-host, Dr. Jimmy DeKiakos, and co-conspirator. You'll be seeing and hearing from him very shortly, and to our three panelists. But before we move on to the talks, I'd like to introduce Dr. Damien Joly, a colleague at the Canadian Wildlife Health Cooperative, who will be providing some opening remarks. So the Canadian Wildlife Health Cooperative, or CWHC, is a collaboration of the 5 Canadian veterinary schools and the BC Ministry of Agriculture. Damien is the CEO of CWHC, and most of his career has really been focused on emerging and re-emerging zoonoses of wildlife origin at the human-livestock-wildlife interface. I should say both Damien and the CWHC have really been at the front lines of this epizootic and panzootic event concerning highly pathogenic avian influenza virus, and it really is in partnership with other federal agencies as well as provincial and territorial partners that they have been meeting this challenge. So Damien, over to you.
Great, awesome. Thank you for that great introduction. I'll just share my screen here. There we go. So hopefully that's working. So bonjour tout le monde. Good morning, good afternoon, everyone. So I'm just going to give some introductory remarks about avian influenza in wildlife and how it came. But before I start, I just want to, pardon me, to acknowledge that I live and work and play on the traditional lands of the Snuneymuxw First Nation on Vancouver Island. So it's just right over here, right? And I walk my dog every day in these woods, and it's really important to me just for my mental health to get out there. And it's wild. Like about 5 kilometers from here are these petroglyphs. These are carvings in stone that have been dated up to a millennia. So providing evidence of continuous habitation by the Snuneymuxw people for millennia, for thousands of years. And it's just a great reminder that every day that I am a guest on these lands, and really honored to be able to live, work, and play on these lands. But it's also important to note that the Snuneymuxw First Nation is not, this is not a nation that existed 1,000 years. They're an active culture that's alive and active today. So I put up their website if you want to check out kind of things that the nation is up to. It's right there. So thank you. I just want to acknowledge that before I move on. So I'm the CEO of the Canadian Wildlife Health Cooperative, and as Dr. Mubarak mentioned, we're a collaborative of the 5 veterinary schools in Canada, as well as the BC Ministry of Agriculture and Food Animal Health Centre in Abbotsford. So we work with our federal, provincial, territorial partners to essentially form the backbone of Canada's wildlife disease surveillance system. So our mission is to promote, essentially to promote and protect the health of wildlife in Canadians through leadership, partnership, and investigation and action. So today I'm going to talk about avian influenza. So avian influenza, the ancestor of the current strain we'll be talking about today, first emerged in 1996 in Guangdong province, and it sort of percolated in the domestic animal populations of Southeast Asia for about 10 years until about 2005, 2006, when it emerged in Qinghai Lake, where there was a massive die-off of thousands of bar-headed geese in Qinghai Lake. And I don't know if you can see my cursor, but there's this little pink spot in northern Mongolia where we had work doing surveillance. We were actually able to isolate the virus, the first folks outside of China to get access to that virus because of the flyway. So the virus spread across Asia, or Eurasia, in 2005, 2006, and then largely died out in wildlife, although it stayed active in domestic populations. So this graph shows the number of reports to the FAO for outbreaks of avian influenza over the last couple decades. And you'll see that in 2005, 2006, there was a big pulse of reports where it spread across Eurasia and then largely died out in wildlife. We'd have intermittent outbreaks in wildlife for about a decade or so, whereas it continued to percolate in domestic animal populations. 2005, 2006, 2015, 2016, 2017, lost a decade there. The rules changed for avian influenza, where it started to actually persist over winter in wildlife populations and spread across the Eurasian continent until 2021, where we saw avian influenza across Eurasia. In 2021, it jumped the pond. We saw our first cases in Newfoundland. They were almost simultaneously cases in wildlife as well as in domestic animal populations. So these are a couple of sort of cartoons showing potential of how this virus might have spread to North America, either through waterfowl populations going to Iceland and then over, or through pelagic birds. We don't actually know how it got to North America, but we do know in the fall of 2021 it showed up. And wildlife had their pandemic, or panzootic as we call it. So the virus spread across the continent, affecting likely thousands, hundreds of thousands of wildlife, as well as domestic animal populations. So we've, just in terms of testing, the Canadian collaboration on avian influenza was able to detect over 1,800 positive bird cases. So these are just birds that were sampled, brought, tested, and tested positive. That doesn't reflect how many birds actually got the virus, just how many we detected, and as well as almost 140 mammals across 7 different species. So this virus is very good at jumping between species, and as it spread across the continent, affected a large number of wildlife. As well, in domestic populations, we've seen so far almost 11 million birds affected by avian influenza across 350 farms, infected premises where folks have poultry populations. So this has been a dramatic economic impact, as well as has had a major impact on wildlife health across the country. So what have we learned so far from this? We've learned that HPAI, since it emerged in 21, has had a dramatic impact on both on our economy as well as on the health of wildlife and domestic animals across the country. Some of the work I've done previously with a project called USAID Predict, we did some research looking at how often, or what are sort of the ecological or epidemiological characteristics of viruses that might increase the risk of pandemic. And one of the things we found is that viruses that are good at jumping between hosts have an increased likelihood of forming a virus that's a pandemic. So this has been quite a concern for us who work on avian influenza, that just to see the vast number of mammals being affected by this virus. The other thing we've learned from this is really important, the large importance of One Health, of working together. The Canadian collaboration on avian influenza is vast. We have all the provinces and territories across the country participating, as well as a number of different federal agencies. So we're on phone calls almost every week, talking about avian influenza, working with folks across the spectrum from wildlife, domestic animal, and human health folks. And this is really a unique collaboration. I've worked in many countries around the world, and to see the collaboration, the degree of collaboration across, these sectors in Canada is just brilliant. And we really wouldn't be able to have a good handle on what's going on with this virus if we weren't for this type of collaboration. So, pardon me. So with that, I'll pass it on, but thank you very much for giving me a chance to talk today. Oh, Erin?
That's great, Damien. Thank you very much. I will jump in. Much appreciated. And thank you for the really wonderful talk. I believe Jimmy is going to jump on here shortly and introduce.
Okay, I'm right here. Thank you very much. I just can't start the video, Erin, but that's okay. I'll just keep going. It is my pleasure to welcome everyone and to introduce our next speaker, Dr. Stacy Schultz-Cherry. Dr. Schultz-Cherry is a member of the Department of Host Microbe Interactions at the St. Jude Children's Research Hospital in Memphis, Tennessee. After a tenured associate professor position at the University of Wisconsin, she brought her pioneering research program on viruses to St. Jude's in 2009. Oh, maybe now you can see me. Hello, everyone. She is an internationally renowned investigator who is also the deputy director of the WHO, the World Health Organization's Influenza Collaborating Center, and the co-director of the NIAID-funded Center for Excellence in Influenza Research and Response, and the NIAID Collaborative Influenza Vaccine Innovation Center. She is also currently a Senior Associate Dean, Academic Affairs Graduate School for Biomedical Sciences at her home institution. Without further ado, I'm pleased to introduce once again Dr. Schultz-Cherry. Go ahead.
Thank you so much, Jimmy, and for the opportunity to speak today. Let's see how badly I can mess this up. Okay, can everybody see that and hear me?
It's all good, Stacy, go for it.
Great, thank you so much. All right, so again, my name is Stacy Schultz-Cherry. I am at St. Jude Children's Research Hospital, and I was tasked with talking about the high-path H5 viruses and really understanding the biology of them. And what I wanted to do, and I want to thank Damien for the nice introduction. I'm going to repeat a little bit of it, but I think it's important to start from the beginning and really understand where these viruses came from, because I think it's important for the biology of what we're seeing now. So I'm sure everybody knows that this really started in 1997 when a series of viruses in mainland China, in quail, goose, and teal, reassorted like flu can do, and started infecting first quail in Hong Kong, and then got into these live bird markets throughout Hong Kong. And it was the first time that we saw that an avian influenza virus could jump species, get into a human, and actually cause severe and even fatal diseases. Before that, we really appreciated that avian influenza viruses could infect people, but we thought of it more as causing conjunctivitis. And this is showing at that time that this proved that this avian virus and this human virus were related, but it really was the only time we've ever seen these H5s, and we knew they were Eurasian descendant viruses. Hong Kong did a great job cleaning up the live bird markets, getting rid of them, depopulated. At the time, I had just started basically a faculty position at the United States Department of Agriculture, Agricultural Research Services, and we were the only people at the time that could really work on these viruses, and that included CDC, because we had the high containment facilities that are required. So we were able to go over there and be involved right from the beginning. And while, you know, it was great that Hong Kong was able to contain this, I guess we sort of knew it was just a matter of time. And that's exactly what happened. Is by 2001, we started seeing these viruses, this particular goose Guangdong lineage virus, starting to move, right? First in Vietnam and then Korea. And what you can see is it very quickly started moving westward, northward, infecting wild birds, domestic poultry, and humans. At the time, there wasn't a lot of reported mammalian infections, and the ones that were reported or because things like tigers in the zoo, other things were fed dead H5 infected poultry. And this virus, again, we've been watching this since 1997. And, you know, it continued to move westward very slowly. You know, here are the different outbreaks over time by the Americas, Europe, Asia, Africa. And, you know, through 2009, this goose Guangdong lineage, you could see it sort of settled over Europe, stayed for a bit, but never really made it across the pond. And then, like Damien said, something happened that it finally did start emerging in the Americas. And this is a really nice timeline of sort of what we know, what we don't know. And, you know, starting with that foul plague outbreak, but then getting into these goose Guangdong lineages. And this is a really nice article, and they're estimating it from literature as well as OIE data of what these outbreaks, 2002, Hong Kong, and then we had China, big outbreak, and then how this just continued to move in these outbreaks helped drive the evolution of these viruses. Because remember, that initial phylogenetic tree with the HA and NA was one virus. And through WHO, we have been watching and renaming the sub-clades coming off as the virus continued to move. And this is a more updated phylogenetic tree of the goose Guangdong lineage H5 viruses. And you can see how many different subclades that we had. And Damien just showed this really nice data out of Hong Kong, really showing where things were, what animal species were involved. And then if you look at the evolution of this virus, I think it's really intriguing in that we had a clade 1, and then we had 2, and then by 2007 we had a 2.2, and this is all based on hemagglutinin. And it took a long time. That 2.2 clade circulated for quite a while, and you would get subclades off of it, and we'd have to update our candidate vaccine viruses. But it really wasn't until about 2013 that you can see there was this sort of explosion of variants coming off. And about that same time, we started going from solely H5N1 to the H5 now picking up different NAs, different internal genes. And all of this really played into the evolution of this virus. And what you can see is the evolution by different countries over time, and then even by countries, and then different host species, and how each region that was impacted by the H5 was impacted by different strains, different mammals were impacted. Again, not a lot of mammals being reported, but every place was dealing with something a little bit differently, except for the Americas, where we didn't have to deal with it at all until recently. And when you look at these maps, one thing you'll notice, so we've got Africa, we have got Southeast Asia, we've got North America. There is nothing about South America. And this is really important when you think about the ecology and evolution of influenza in the Americas, is these flyway zones, where we know these birds are wintering in South America and really converge all 3 flyways within South America. And this is actually where we do a lot of our field work in both Chile and Colombia. And so we've been at the forefront. We've been watching this thing come for years, having really started a lot of field surveillance in South America in about 2013. And so you can see what began happening about October 2022. Things spread in March 2023. And it's even more extensive here, and you can see the number of countries that are impacted. And I'm sure everybody has seen these really heartbreaking images of the ecological disaster that is happening throughout South America. And this is an image that one of our colleagues from Santiago, Chile sent, and you can just see this pelican. and you immediately know it's either velogenic NVB or type-bath flu. And so we were the first to sequence and report on the H5 goose Guangdong lineage in Chile. And you can see here, it's a North American lineage HA. So this came down likely probably the Mississippi flyway based on the closest genetic relatives. And we're really concerned about this because there's a lot of avian influenza circulating in wild birds throughout Chile. And this is just showing a map. And we know we're in a unique situation there in that we're the only country that we know of right now that has the goose Guangdong lineage co-circulating with low path H5. And so that is a reason for concern. And we're very worried about this, is that in these wetlands throughout Chile, where there's a lot of interface between domestic birds, wild birds, a lot of backyard production systems, a lot of companion animals and people that are just the right sort of breeding ground for this to happen. These are some of our field workers. So we're doing a lot of work there on surveillance in all of these, including at the animal-human interface. We're also watching the situation in Colombia. That's another area where we do a lot of studies. And we know this is the entry and exit point when birds are coming from North America into South America. And it's usually right up in these regions of Colombia. And they are seeing H5 in wild birds. And then, of course, as they start looking and doing surveillance in domestic poultry, they're finding it. We have studies, ongoing human cohorts in indigenous populations throughout that Caribbean region of Colombia. And we are very concerned about this animal-human interface because it's kids, they're a non-vaccinated population. It is not, you can see what we're seeing in some of these villages are just dead birds. Here's an image from Santa Marta. And it's these kids picking up these dead and dying animals, plucking the feathers, and then of course they eat them. That we're now worried about this population being at this interface. So what happens when we identify an H5 or any other virus out in the field? Well, there's a number of things that we want to know, particularly about, is this a zoonotic risk? What host factors are involved? Do we have zoonotic transmission? And then even getting at from the animal, the interface, the virus, and to in those people, what are we seeing? And we take all these viruses and we put them through virus characterization pipelines. And we really look for ones that come to the top of the surface. So anything antigenically novel in H1 and H3 and H2, anything that is spilled over, any avian that has spilled over into mammals, raises a concern. And we look for things like molecular determinants of virulence, receptor binding specificity, pathogenesis in animal models, transmission in ferrets, population-wide immunity. And then we use this information to inform and sort of predict risk of emergence and then risk of sort of prevalence. And we use both the Centers for Disease Control as well as WHO's risk assessment pipelines. And then this information through our WHO collaborating center gets fed to all of the proper authorities, including WHO, and it gets into GISRIS. So this is all great. We do a lot of field work, but my laboratory is really very much fundamental basic research, and we want to understand influenza viruses and susceptibility. Who gets infected? Why do they get infected? And then what happens? And what's associated with that severe disease? How is the virus really regulating this? And how do we treat it? And as I mentioned, being at USDA, we were amongst the first to really be able to start characterizing these goose Guangdong lineage H5 viruses. And so I'm going to tell you just a couple little really sort of unique biological properties of these viruses. The first being that when we looked at some of those original strains And one thing we found is they had a very different tropism. So of course they infect your lung epithelium. They are really good at destroying nasal epithelium. And these viruses, because of that alpha-2,3 sialic acid linkage, can very rapidly get deep into the lungs. And I'll show you why this becomes important. But another thing we found that we didn't expect are that these lineage viruses can productively infect macrophages, which we've never seen before. And so these are using human primary macrophages as well as primary murine alveolar. What you can see is when you infect them and take just a single time point, only the H5s, and it's only these goose Guangdong lineage, can productively infect this macrophage. And we thought, well, maybe it's the representative strains that we're using for each. So then we looked within some of these, a bunch of different strains. And again, we found that it's these goose Guangdong lineage. If you look at some other low path H5s, you look at some of the older H5s, high path, we just don't see it. And this is sort of a really nice image over time of human macrophages infected with California 9 human virus, or one of these H5s. And you can see that these actually don't just infect, they actually replicate. And this is true in your alveolar macrophages. And this becomes very important in the pathogenesis, because what ends up happening is they're replicating and they're not necessarily killing these macrophages. We do see some cell death, as you can see here. More importantly, they are completely inhibiting the function of the macrophages. So you can imagine how troubling this is going to be when you get into pathogenesis. The other thing they do is they're very, very good at infecting your alveolar epithelial cells and reducing, even if they don't kill the cells, again, they interfere with that alveolar function. So they don't allow those alveolar cells to clear fluid, and they increase the barrier permeability, which means you end up with leaky lungs, you get into acute lung injury, and you get into acute respiratory distress syndrome. We see this in mice, we see it with some strains in ferrets, and we'll talk about that in just a minute. And this is what was reported in some of the humans that were infected. The other thing it can do is productively infect your endothelial cells. No other strains of influenza that we have found to date can productively infect different types, or even really any types of endothelial cells. And so it makes sense that when these get into an animal, and we'll start with a mouse, they can cause very severe disease. So if you do a mouse lethal dose 50 experiment, different titers, you even compare it to some of the highly pathogenic H7 viruses that emerged later in China. The H5s, the goose Guangdong H5s, are always worse. They're always more severe disease. They cause more inflammatory responses. I hate using the word cytokine storm, but in this case, it's probably a fair statement, is that you can see is that if you look at Vietnam 1203 or Hong Kong 213, some of these early H5s, when you look at interferon gamma in the respiratory tract or in the systemically in the plasma, they are really driving a very potent inflammatory response. And that is very evident when you start looking pathologically. At least with these older viruses, most of this is mediated by the hemagglutinin itself. So back in the day, before gain of function, studies were done where you could take Hong Kong, a goose Guangdong H5HA, put it in California 9, use it in the high containment facility, and you always see that it's much, much worse. The NA also has problems in that it doesn't activate a very special growth factor that is required in your lungs for protection from flu. So these are two different proteins where we know that there are differences that are associated with pathogenicity. And so we've looked at some of the mammalian, the old goose Guangdong lineage viruses that did come out of mammals. So in this case, this was in Cambodia. Bears in the zoo were fed H5 infected birds. Bears got sick. bears died. Here's your phylogenetic tree. What you can see is that these bear-derived viruses grow very well in MDCKs, as well as primary normal human bronchial epithelial cells, basically the same way a California 09 virus would. And it's even this avian virus replicates incredibly well. And when you get in with these bear viruses, we also had otter. Didn't matter if you got the virus from the bear's brain, the other end of the body, they were across the board lethal in mice. So let's think about these newer viruses. What's happening now? What do we know about the new high-path H5 Busquandong? Well, we know in North America and the US, they are already reassorting. And so this is a really nice paper from Richard Webby here at St. Jude, where it sequenced a bunch of different isolates. You can see that the genotypic differences. In a mouse, it does exactly what we would expect, kills them. In a ferret, typically most of these H5 strains don't do much, but in this case, some of these strains are lethal in these ferrets due to really high systemic virus levels, and due to the fact that some of them, it's not due to lung and pneumonia, but a number of these viruses are going straight to the brain. And we don't know, we're speculating that it's due to that olfactory, upper respiratory tract infection going up the olfactory bulb, more like an H7 virus can do. So what do we know about what's happening in South America? We've got these strains down there now. We have different circulating viruses in the environment. We know we're getting the viruses up now. We know they're already starting to reassort. And the answer is nothing yet. But ask me in about 6 months and we'll be able to give you a different answer. And with that, I want to thank all my lab and just thank you guys for listening and just show you an actual image of a mouse infected, flu infected lung. Everything in blue is actually influenza virus. And this is a real lung in 3D. So thank you very much.
Thank you so much, Stacy. That was really wonderful. And I'll just point out that there is a question in the Q&A, and either you can answer it directly there or we can come back to it at the end as well in case you want to elaborate on that to the group. So thank you very, very much for that. Much appreciated. And it's my pleasure now to introduce Dr. Bryce Warner. So Dr. Warner completed his Bachelor of Science and his Master's at the University of Saskatchewan in Saskatoon, and then his PhD at the University of Manitoba in Winnipeg. So he worked on Cynombre virus in Peromyscus or deer mice, and this is quite notable because Cynombre is one of the few high consequence viruses that are actually endemic to Canada. And he's currently a biologist in the Special Pathogens Group at the National Microbiology Laboratory in Winnipeg. His research focus has been on developing animal models for high containment viruses, and these include hantaviruses, arenaviruses, coronaviruses, and as you're just about to hear about as well, highly pathogenic avian influenza viruses. So really focus on animal models as well as the development and testing of medical countermeasures, including vaccines and therapeutics. And most recently, this work has been involving high-path avian influenza, as I mentioned, but also specifically around risk assessments of the various mammalian species, or for various mammalian species, with the specific Canadian viral isolate. So over to you, Bryce. Thank you.
All right. Thanks for the intro, and thanks for having me to speak. So I'll just thank the previous speakers for covering all the basic introductory stuff, so I can just jump right into our data. But I'm going to talk a little bit about what the previous talk. I'll just expand a little bit about that and discuss sort of the risk assessments we've been doing with our collaborators at CFIA and the domestic and wildlife samples that they've been getting and the isolates they've gotten, and what we've been doing on our side with sort of basic laboratory characterization in mammalian species. So this slide really just is an overview of the isolates I'm going to discuss in detail. So there's 5 here highlighted in red, just with the species that they were isolated from. So 2 from red foxes, 1 in PEI, 1 from Ontario, 1 from a red-tailed hawk, and 1 from a blue-winged teal, and then 1 from a turkey. And as was mentioned, after the viruses, sort of made their way into Canada, into North America, there's of course been reassortment, and so different reassortment patterns of the viruses that we've been working with I've just shown here. So the red fox PEI is a fully Eurasian genome, and then 3 of the viruses have this second pattern here. So with PB2, PB1, PA, and NP of North American origin, and the other segments of the Eurasian Goose-Guangdong lineage origin. And then the red-tailed hawk virus has this other pattern 5 with PB2, PB1, and NP of North American origin. And I'll just quickly mention too that this is the 627K of PB2 is in the red fox and the red-tailed hawk. The red fox Ontario has a valine at the residue, and then these 2 avian viruses have the glutamic acid, which is of note. And then I just wanted to put this up quickly for reference that, when you do a consensus, so these are the 4 isolates that we did our initial risk assessment and characterization in mammals. So the ones I'm going to discuss right now, if you compare sort of the consensus sequence of all of the proteins, there aren't that many differences. And so the one I'm going to focus mostly on today is the one from red-tailed hawk. And what's interesting is there's only 6 unique amino acid residues to that virus when you compare it to the other ones that we've looked at. And one of them in HA, 4 in the PA, and 1 in NS1. And so I think it's interesting that what you'll see is that the phenotypes are quite different, even though genotypically they're quite similar. And I appreciate what Stacy showed earlier with some of the LD50 data in mice. So I'm just going to quickly show what we did in mice with these 4 viruses. We did an LD50 with each of them in BALB-Cs, and you can see from this table here that the LD50s are quite low. So for red-tailed hawk virus and the red fox Ontario, we still saw 100% lethality even when we went down to 1 PFU. And then for the red-tailed hawk virus, you can see even within 5 days, so very high virulence with this virus in BALB-Cs. And then because we have a deer mouse colony at our disposal here for hantavirus work and other things, we did put some of these viruses into deer mice as well, just out of curiosity and to see, because they're a highly abundant mammalian species in North America, what might happen. And what was interesting is that, at a high dose, it was, uniformly lethal, again, for the red-tailed hawk virus and for this red fox Ontario virus, very fast lethality, again, for this virus here. But for the red fox PEI, the one that was wholly Eurasian, there were no lethalities in the deer mouse when we did this. So it's interesting, and it indicates maybe that the segments may be playing a role, and these are North American rodents as well. But I'll jump right into the human and ferret work. So we did initial characterization, our collaborators did, in human airway, air liquid interface culture, so primary human airway cells, so bronchial, tracheal cells, small airway cells, and nasal epithelial cells. And what you can see is quite high replication very quickly for some of these viruses, particularly the red-tailed hawk virus in gray here. So to a very high titer, very quickly. And you can see that here in the IHC staining as well. Has clearly the strongest staining for sure, at least in the bronchial tracheal cells as well. But some of the other viruses are also able to replicate to quite high titers in the human airway cells as well. But we went ahead with these initial 4 isolates that I've shown here with a ferret transmission experiment. And so initially we had 18 ferrets for this. And so this is how it was set up with a direct contact experiment with 1 or 2 infected ferrets in a cage, and then a naive contact ferret added in a day later. And so we give 10 to the 6 PFU intranasally, and then a day later added the naive animal, and then just monitored the animals' daily weights, temperatures, and then we did swabs every other day just to examine viral shedding. And so what we found from this initial study was all 4 viruses were virulent, and they led to death in most of the animals. There was only 1 survivor here with the blue-winged teal virus, but all 3 other viruses were uniformly lethal. The red-tailed hawk virus and the red fox Ontario quite fast, quite a bit faster. So, but what really stood out in this initial characterization of these viruses was that the contacts for the red-tailed hawk virus also died, and none of the contacts for the other 3 viruses did it. And when you look at the weight loss, you can see the trend. They lose weight quite rapidly, and then as well in the contacts too. And then when we looked in the tissues, these are sort of the log 10 titers in all the tissues that are listed. So pretty good systemic replication in a lot of the animals, and then also, you know, neurotropism and brain infection in about half the animals that we looked at. And we've done subsequent experiments where, you know, infection of the brain has been quite prevalent as well. And then just focusing on the red-tailed hawk virus, again, the one that had transmission to the direct contacts, you know, you can see why that was the case. You know, when you look at the 3 infected animals, they shed very high levels of infectious virus in swabs, in both the oral swabs and the nasal swabs. And so, you know, it makes sense that those contacts would end up being infected, and then, you know, they went on to require euthanasia as well. So we wanted to follow up with that specific virus, the red-tailed hawk virus, just because, you know, even direct transmission, direct contact transmission with these viruses is not that common. And so, you know, we thought maybe this is something unique happening with this virus, and it was of interest to us. So what we did was we set up a similar study with an infected animal per cage with a naive direct contact again. But this time we used the double wire mesh barrier between one other animal. So this was the airborne contact. This one was added in a day later as well, with airflow going in this direction. So potentially, if there's respiratory droplet transmission, that might be detectable using this system. And again, the same monitoring of weights, temperatures, and sampling as well. And so all the, you know, relevant data is here essentially. So what we found again was very fast time to death in the infected animals, 2 on day 4 this time and 2 on day 5. There were 12 animals in total for this, 4 in each. And then out of the direct contacts, 1 animal had to be taken down on day 6, and then the experiment ended on day 8. But when we look at the direct contacts, you can see this one here, it was losing weight quite quickly and had to be taken down. These other 2 were trending down quite quickly, and you know, if the experiment had extended a couple more days, these animals would have had to be euthanized as well. No clinical signs were detected in any of the airborne animals, so that it sort of indicated there was no airborne transmission. When you look at the temperatures, though, you can see all the infected animals had high fevers, as well as the contacts. All the contacts that we know ended up being infected had high temperatures. And then when we looked at the tissue titers at day 8, when we ended the study, again, high titers in most of the tissues, including systemically and in the brain of some animals as well. And then the 3 contacts, and then this contact, the one that didn't have any clinical signs, no infectious virus detected. And then in the 4 airborne contacts, no infectious virus in any of the tissues that were sampled. However, when we ran PCR on some of the tissues, including nasal turbinate and lung samples, they were PCR positive, and these are just the CT values here. And so this indicated that maybe, you know, there was that these animals might have been infected. We didn't leave them long enough to see any clinical signs, or, you know, that we ended the experiment too early. That maybe a little bit more time for replication, we would have seen maybe some infectious virus in these tissues had we waited a little bit longer. But we know that, you know, if we have CT values sort of in this range, low 30s, high 20s, low 30s, you know, the MDCK TC8050 assay may not be sensitive enough to pick up infectious virus, but if we inoculate eggs, we might be able to show that there is replicating virus in there. And so that's what we did with some of these tissues. So we had our tissue samples that were PCR positive. We made single cell suspensions and homogenized them. Took supernatant and filter sterilized and inoculated special pathogen-free embryonated eggs with each of those. And then we candled them every day for 3 days. And then any eggs that we saw embryo death, we would collect the allantoic fluid and then at the end of 3 days, we collected the allantoic fluid of all the eggs regardless, and then we went forward and did TCID50s and PCR with those to see if there was replicating virus in any of the samples. And so this table just sort of summarizes that. So all of these samples here that I mentioned before were all MDCK TCID50 negative, and these were their initial CT values. And then following egg inoculation, an increase in virus in almost all of the samples, except for a couple, but most of them, a CT value from low 30s down to 11, 15, 17, etc. And then importantly too, most of these samples, all of them except for 2, were positive by MDCK TCID50 assay. So that indicated that there was virus present in the tissues of some of these animals, despite the fact that we didn't see any clinical signs in this experiment in the airborne and the one contact animal. So the conclusions just from this initial work, our initial study showed, again, high virulence for all the viruses tested, particularly the hawk virus in the Red Fox, Ontario, lethality within a week, high infectious virus in all the tissues, including the brain, direct contact transmission leading to death in the red-tailed hawk animals, and then including the follow-up, again, direct contact transmission in 3 out of 4 animals, rapid lethality, high virulence, and then the potential airborne transmission we detected, via the egg inoculations with the tissues from the PCR positive animals there. So future and ongoing work. So we've done a bit of follow-up with the red-tailed hawk virus in some airborne transmission studies. We've also done some additional studies that I didn't share, that I'm not sharing today, just for sake of time, with a handful of other isolates from turkeys, from skunks, and then one from an H5N5. Lots of these viruses have similarly very high replication. So I think, there are many more isolates that are of interest that replicate well in human airway cells and in mammalian species. Again, these also have shown high virulence and direct contact transmission in ferrets, and potential airborne transmission that we're following up on as well. And then we, like in Stacy's talk, where she mentioned a whole myriad of factors that play a role. We're assessing sort of the molecular and genetic determinants of this transmission and virulence, and the sort of the differences that we've seen between the viral isolates that we have. So I'll just quickly thank everyone in the lab that I'm in, Darwin Kobasa's lab at the National Microbiology Lab in Winnipeg. This is our group here. And then our collaborators at CFIA, Johannes and Tamaru, who provide us with all the viral isolates they receive and do all the air liquid interface culture work, and then our collaborators at Sick Kids and Sunnybrook in Toronto. So thank you, and I'll I guess take any questions at the end.
Excellent. Thanks, Dr. Warner. So I have the great pleasure and honor of introducing Dr. Matthew Miller as our next speaker. So Dr. Miller is Executive Director of the Global Nexus School for Pandemic Prevention and Response and the M.G. de Groot Institute for Infectious Disease Research at McMaster University. He also serves as co-director of the Canadian Pandemic Preparedness Hub, CP2H. Dr. Miller holds a Canada Research Chair in Viral Pandemics and was recruited to McMaster University in 2014 as an assistant professor and has authored over 80 peer-reviewed publications. He's received several major awards, including a CIHR New Investigator Award, the CIHR Bhagirath Singh Early Career Award in Infection and Immunity, and an Early Researcher Award from the Government of Ontario, as well as a Distinguished Alumni Award for Excellence in Basic Science from Western University. Dr. Miller's research program is focused primarily on pandemic prevention, especially the development of broadly protective vaccines, antibodies, and therapeutics. He sits on the scientific advisory board of several major vaccine manufacturers, including Seqirus, Pfizer, and Sanofi. He's a member of the Canadian National Advisory Committee on Immunization, COVID-19 Working Group, and Committee to Advise on Tropical Medicine and Travel, the Federal Variants of Concern Leadership Group, the Federal Pandemic Science Coordination and Action Group, and the PHAC Expert Panel on Highly Pathogenic Avian Influenza. He's served as Secretary Treasurer of the Canadian Society for Virology from 2021 to 2022. So I'm honored and pleased to introduce Dr. Miller, and I'm really looking forward to hearing your talk about HPAI vaccines.
Well, thanks so much for that introduction, and I really appreciate the invitation to speak to all of you today. My presentation is, as was noted, going to focus on highly pathogenic avian influenza vaccines, in particular vaccines for humans specifically. So I'll see if I can advance my slides here. Here we go. So unlike the situation we were in during COVID-19, where prior to the COVID-19 pandemic, there were no approved coronavirus vaccines for use in humans, with influenza virus, we have an almost unprecedented number of different vaccine types approved for seasonal flu. These include live attenuated vaccines. Some of you may know this as FluMist, recombinant vaccines. This is hemagglutinin that's produced in insect cells. There are whole inactivated vaccines available in some jurisdictions, though not in Canada, as well as more conventional split and subunit vaccines. And as all of you can imagine, there's a ton of ongoing work in the flu field, like in almost all other fields of vaccinology, using new platforms and technologies to generate new and better vaccines, including things like mRNA vaccines, for example. In addition to all of those different vaccine types, we produce influenza vaccines in different substrates. So classically, influenza vaccines have been produced in embryonated eggs. And although the majority of seasonal vaccines are still produced this way, there's been sort of a general push by the field to try and move away from egg-based vaccine production. The benefits of producing flu vaccines in eggs is that you can make a lot of virus. It's very efficient typically. But one of the major problems is that in certain cases, the vaccine strains can adapt in eggs in a way that changes their antigenicity. And in doing so, creates mismatch with the strains that actually circulate. And we've observed on many occasions that that can significantly decrease vaccine effectiveness. In addition, there are now cell-based vaccine platforms. These vaccines have the benefit of moving away from egg-based production, and as a result, you don't see the accumulation of these egg-based adaptations that can occasionally lead to antigenic mismatch. And we also have these recombinant vaccines, which are purified hemagglutinin molecules produced using the baculovirus system in insect cells. What's also shown in this nice figure is the timeline of influenza vaccine production. And you can see that one of the things that's a real challenge in producing seasonal flu vaccines is that the WHO meets and makes recommendations about which strain should be included in the upcoming Northern Hemisphere vaccines in around February. And then in March, this vaccine production cycle begins, culminating with vaccines being available in October. And one of the challenges is that if the predominant circulating strain changes in a way that is antigenically important in the intervening months while vaccine production is happening, there's basically no opportunity to go back and correct that vaccine. And that can also occasionally lead to mismatch. Now, despite the vast number of flu vaccine platforms that are approved and available, if you look in aggregate at seasonal flu vaccine effectiveness across seasons, what you'll note is that at best, seasonal flu vaccines are about 60% effective. And so while that's great, it does lead to really meaningful protection and reduction in circulating flu. It's certainly far from optimal. So even in the seasonal space, there's a lot of room for improvement in terms of increasing vaccine effectiveness and efficacy. What you can also see here, as I noted in an earlier slide, is an example of how vaccine effectiveness can be impacted by egg-based adaptations. So in 2014, 2015, there was an important, well-known egg-based adaptation that happened in the H3N2 component of the vaccine. And as a result, vaccine effectiveness was much lower than what we typically observe. Now, the most recent influenza pandemic, as many of you will remember, was the 2009 H1N1 swine flu pandemic. And again, in contrast to what we've just experienced during COVID-19, in 2009, we had a massive global infrastructure capable of producing flu vaccines. We knew how to produce them. There were distribution pipelines in place. And yet, when this 2009 swine flu pandemic emerged, the seasonal vaccine for that season in the Northern Hemisphere was already in production. And so what happened was that manufacturers went back and made a monovalent vaccine specific to this new 2009 H1N1 swine flu virus. What you can see in this graph, though, is the realities of reactive pandemic vaccine production. So here in blue, the graph is showing the incidence of influenza-like illness during the pandemic. In red, you see the number of vaccine doses shipped, and then in green, the number of those doses administered, where there's obviously a lag. And what I hope you can appreciate from this is that even in a situation that was in some ways optimal in terms of our ability to produce and distribute a vaccine, we were still way behind the curve in terms of being able to get vaccines out to people. Thankfully, this was a relatively mild pandemic, as pandemics go, but you can imagine that responding in this way for a future serious influenza pandemic would result in huge amounts of global morbidity and mortality. And so we really need to change the paradigm of how we address pandemic vaccines for flu. In Canada, the Canadian government awards contracts to influenza vaccine manufacturers to produce pandemic flu vaccines in the case of a future pandemic. The current contract is awarded to GSK for their pandemic vaccine platform, and the agreement is to produce up to 80 million doses in the event that a pandemic's declared. The GSK production facilities in Saint Foy, Quebec, which also makes seasonal vaccines for the Canadian population. The GSK vaccine is an ASO3 adjuvanted split inactivated vaccine. So it's a very conventional vaccine platform with the addition of an adjuvant, which augments the immune response and the immunogenicity, obviously. Both monovalent H5N1 and H1N1 formulations have previously been approved in Canada. However, I think it is important to note that GSK is not the only manufacturer of approved pandemic vaccines. Almost all of the major seasonal influenza vaccine manufacturers have these types of products in their pipelines, and some also have language built into their seasonal vaccine procurement contracts that they would provide pandemic vaccines to Canada if a pandemic were to occur in the future. My real mission, though, has been to try and move us away from the reactive approach that we typically take to pandemic vaccines to a more proactive approach. And in the field, the desire to move in that direction has resulted in the development of several what are called universal influenza vaccine candidates. This phylogenetic diagram on the left basically shows you the diversity of hemagglutinins found in influenza virus. And as you can see here, there are 18 subtypes of hemagglutinin, which is sort of the major focus of the protective antibody response against flu. So that's an immense amount of diversity. However, of all of these hemagglutinins, really only a very small proportion of these viruses have ever circulated in humans efficiently. Really only H1s, H2s, and H3s as far as we know. Of course, there have been periodic outbreaks with other hemagglutinin subtypes, H5s, H7s, and others, but H1s, H2s, and H3s, as far as we know, are the only ones to have caused widespread circulation or previous pandemics. Trying to create a vaccine that's able to cover all of these hemagglutinins would then seem like a very daunting task. However, what you can see if you start looking left on this phylogenetic tree is that within influenza A viruses, this tree bifurcates back here where I'm showing with the red cursor. That bifurcation, what it actually represents is conservation in what's called the hemagglutinin stalk domain. So the sequence of hemagglutinin that leads to all of this diversity, that sequence diversity is mostly found in this highly variable hemagglutinin head domain, which is extremely tolerant to mutations. However, the stalk domain is much less tolerant to mutations and much more conserved. And as a result, there's been a huge amount of research and attention focused on generating vaccines that would produce antibody responses against this highly conserved stalk domain. And in doing so, is hoped to provide more broad protection against influenzas, including highly pathogenic avian influenzas. The discovery of the strategy with which to produce these antibodies in high titers was really defined in the wake of that 2009 H1N1 pandemic. In typical situations, what happens the first time an individual is exposed to influenza is that they make high titers of antibodies against this antigenically diverse head domain and relatively few antibodies, against this highly conserved stalk domain. The reasons for the immunodominance of the head domain are becoming better understood, but I'm not going to get into them today for the sake of time. Now, in typical situations where flu drifts seasonally, what happens is that relatively few mutations within the head domain lead to a reduction in the amount of neutralizing antibodies that recognize the virus. However, most of the hemagglutinin molecule itself is left unchanged. And as a result of that, what happens when you're exposed to a new strain of flu is that you boost a lot of the antibodies that are non-neutralizing or are weakly neutralizing that were specific to strains you've encountered earlier. You still don't make very many of these antibodies against the stalk domain. And then, of course, you make a de novo response to the new epitopes, the new mutated epitopes in the new strain of virus. What happens during a pandemic, though, is that the head domain of hemagglutinin is typically so foreign that you don't mount an appreciable memory response. And as a result of that, you're left making this de novo response to a new head domain. In that situation, the one thing that a previously exposed individual's immune system can recognize is this conserved stalk. And we now know that it's in these situations that you're able to induce high titers of these broadly protective antibodies that bind the stalk. When I was a postdoctoral fellow in Peter Palazzi's lab, our group did a lot of work using this information to develop a universal vaccine candidate based on what are called chimeric hemagglutinins. These are hemagglutinin molecules where we can essentially genetically stick on any head domain of hemagglutinin we want on a conserved stalk. And using that strategy, we were able to build vaccines, both sort of conventional split or subunit vaccines produced in eggs, as well as live attenuated vaccines bearing these hemagglutinins. Both Peter and Florian Kramer, who is also a postdoctoral fellow when I was in Peter's lab and now has his own very successful group at Mount Sinai, conducted a clinical trial in partnership with GSK to test this strategy. And what they found is that if you sequentially immunize individuals who have previously experienced seasonal H1N1 viruses with these chimeric hemagglutinins, you mount antibody responses not only to the immunogens that the individual receives in the vaccine, but also you generate high titers of these broadly protective stock antibodies that can recognize hemagglutinins that have never been encountered before. In collaboration with Rafi Ahmed's group, and this was a study led by Ali Edebedi when he was a postdoc in Rafi's lab. Ali's now has his own, again, very successful group at WashU. We did a study where we immunized individuals, adults, with either a seasonal influenza vaccine, a typical seasonal influenza vaccine, or a monovalent H5N1 vaccine. And what you can see here are the domains of hemagglutinin where the antibody response is focused. So as I alluded to earlier, this is sort of the typical situation. You give someone who's experienced H1 viruses before, either through vaccination or infection or both, a seasonal flu vaccine. They mount really strong responses against the head domain of hemagglutinin, but basically they don't boost stock responses at all, as you can see also shown over here in this scatter plot. However, something really interesting happens when you immunize with the H5 vaccine. When you prime individuals, what you can see is there's almost no neutralizing antibodies made against the H5 head, and yet you mount these really strong responses against the stock. And of course, the reason for this is because the stock of H5 and the stock of H1 is very closely related. And so there's immunological memory to these epitopes, whereas there isn't initially to the head. However, what I think the coolest part of this study is that if you bring these same individuals back a month or so later and boost them with the same virus, now the antibody response shifts back to the typical situation. In this prime, they've mounted a de novo response to the head. And so upon subsequent exposures, the preference of the immune system shifts back to recognizing these neutralizing epitopes in the head domain at the expense of these stock-specific responses. Now, in trying to create broadly protective vaccines, both in the flu field, but certainly also in the HIV field, I'd argue that we've historically taken a very antigen-centric approach. We think really hard about finding conserved parts of the virus that we can target in order to mount broadly protective responses. And while that's certainly a very important consideration, I would argue what we've thought much less about is the way in which we administer vaccines. Many of you will know that mucosal vaccines are something that's near and dear to my group's heart. But what is really important about the route of vaccine administration is that it profoundly changes the quality of the immune response. So when you inject a vaccine intramuscularly or intradermally, what you mount are typically very strong systemic antibody responses that are typically dominated by IgG, as well as, in some cases, circulating T cells. However, if you immunize someone mucosally, that response shifts to an antibody response that's typically dominated by IgA, particularly secretory IgA, tissue-resident T cells, as well as induction of trained innate immunity, which I'll get back to in a couple of slides. A lot of the pioneering work in this field was done by my colleagues at McMaster, Zhou Xing and Fiona Smale, who've been studying inhaled mucosal vaccines in the context of tuberculosis for decades. And a couple of years ago published what I think is this incredibly important landmark study where they used an adenovirus to deliver a TB vaccine either by inhaled aerosol or by conventional injection. And what they found in these human participants was that, well, the injected vaccine generated strong systemic responses, again, characterized by IgGs and circulating T cells, as we would expect. It didn't generate any immunity in the lung, whereas the inhaled vaccine generates both strong immunity in the lung as well as systemic immunity. Based on this knowledge, our group and Dr. Zing's group, as well as Dr. Smale, Dr. Lichty, and many other people at McMaster, collaborated to use this knowledge to develop a next generation inhaled vaccine for COVID-19. And what we found when we did this was very similar to what Zoe and Fiona had reported initially, which is that when the vaccine was inhaled, much lower doses were needed to protect. The protection extended against all variants of SARS-CoV-2 up to and including the most recent Omicron variants. And the intranasal vaccination was characterized by not only antibody responses, but also tissue-resident T cells and trained innate immune responses. What's interesting about trained innate immunity is that it acts much like a traditional adaptive immune response, but in a way that's pathogen agnostic. So these trained innate immune cells, when only trained immune macrophages are induced, because we use animals that are B cell deficient and in whom we deplete T cells, we can still protect completely from lethal SARS-CoV-2 challenge. What's particularly interesting, though, about the qualities of trained innate immunity that are mediated by these airway macrophages is that they don't tend to result in diminishment of viral lung titers. What they seem to do is really coordinate the immune response so that it doesn't result in symptoms or clinical signs. So it's a pathogenicity attenuating vaccine as opposed to a sterilizing vaccine. We've gone on to show that this immunization with our inhaled COVID-19 vaccine can also completely protect animals from lethal influenza virus challenge, even though they've never seen influenza virus before. But consistent with what I told you with COVID-19, we don't see a reduction in viral titers relative to naive animals. Nevertheless, all of the animals survive. Based on this, we've gone on now to develop both a universal influenza vaccine candidate that targets all influenza A and B strains, as well as a specific H5N1 vaccine candidate that targets the clade 2.3.4.4b viruses that are currently circulating ecologically. Now, children are obviously a really important group when it comes to influenza because unlike COVID-19, they are at extremely high risk of severe outcomes. And unlike adults, whom when they get a seasonal vaccine mount very, very specific immune responses, we were also able to use samples collected in a series of really elegant clinical trials run by my colleague Mark Loeb to demonstrate that in children who have these very sort of naive immune systems, sequential vaccination with seasonal vaccines actually does quite a good job of inducing broadly neutralizing antibodies capable of recognizing a much more diverse array of viruses than typical seasonal vaccines are able to induce immunity against, including H5N1 viruses. So this tells us that sequential vaccination of children is a good way to generate broader protection. Finally, what we sometimes, I think, fail to think about is passive immunity or monoclonal antibodies as sort of a complementary therapeutic modality that go hand in hand with vaccines. These antibodies can be used either prophylactically in theory or therapeutically. The broadly neutralizing antibodies that would be of most use for combating influenza tend to work in a way that's very different from conventional neutralizing antibodies in that they talk to cells of the innate immune system and flag infected cells for elimination rather than directly neutralizing virus prior to the virus entering host cells. This is mediated by Fc-dependent effector functions, as you can see here. And what's interesting about these Fc-dependent effector functions in the context of flu is that they rely on the hemagglutinin molecule of the infected cell interacting with sialic acid residues on the immune effector cell. Ali Zhang, a really talented former MD PhD student in my lab, was interested in whether NA inhibitors might be able to synergize with broadly neutralizing antibodies to enhance protection. And the rationale for this was that these NA inhibitors basically prevent neuraminidase from cleaving this interaction, which we know stabilizes the immune synapse between the effector cell and the target cell. And indeed, that's what Ali found. When you combine Tamiflu, or also Tamivir, with broadly neutralizing antibodies, you substantially increase the amount of antibody-dependent cellular cytotoxicity that those antibodies induce. And this is able to protect animals against lethal H5 challenge completely, whereas either antibody alone or also Tamivir alone does not. Lastly, we've been working on aptamers as antibody alternatives. Aptamers behave just like antibodies, except for that they're based on nucleic acid sequences. And the benefit of aptamers is that they can be produced much more quickly and cheaply than antibodies can. During the COVID-19 pandemic, we worked with my colleague, Yingfu Li, who's an expert in functional nucleic acids, to develop a trimeric aptamer that bound to the head domain of spike on SARS-CoV-2. This aptamer was a broadly neutralizing aptamer that recognizes all variants of SARS-CoV-2, again, up to and including the more recent Omicron variants. And really interestingly, what we found is that if we treated animals with this aptamer, the aptamer could protect animals from lethal SARS-CoV-2 challenge just as well as one of the monoclonal antibodies that received emergency use approval. So with that, I'm going to finish up just with a few thoughts on how all of this science needs to marry with policy in order to optimally protect people from HPAI. So some of the outstanding questions we need to think about is, should we vaccinate people against highly pathogenic avian influenza? And if so, who? Should we vaccinate based on occupational risk? So individuals like poultry workers or veterinarians or wildlife workers. When should we vaccinate? Should we do this preemptively? Should we use ring vaccination strategies where we vaccinate contacts of individuals with known exposures? Should we mass vaccinate? And under which conditions should each of those strategies be considered? How should we vaccinate? So in the context of a pandemic, obviously it would be great if you could have a one-shot vaccine, right? But what we know from the previous work that we've done is that with a single shot, you don't make very good neutralizing antibody responses against the specific strain of virus that you're vaccinating against if it's an H5. And so in order to get optimal neutralizing antibody responses, it's very likely that you would need a two-shot vaccine. And finally, what kind of vaccine is optimal to protect against HPAI? Should we stick with conventional parenterally injected vaccine? Should we use inhaled or other types of mucosal vaccines? And what's the place of other complementary prophylactic strategies like monoclonal antibodies or passive immunization? With that, I really want to thank all the individuals who did this work. It's a pleasure for me to be able to present it to you, but my lab are the ones who've actually been slaving away behind the scenes to do the great work I was able to share with you today. So thanks very much. I'll pass the floor back over to the moderators and let us move into the questions session.
Thank you so much, Matthew, for that great talk. If all the panelists can turn on their cameras, if possible, we'll get to the question and answer session here. I'll read the first question out. It's for Stacy's talk. A very interesting presentation. Do you know which 2.3.4.4b H5N1 genotype or reassortment pattern is circulating in South America? That's the question.
So the answer is we don't know yet. We're doing that sequencing now. The original virus that we sequenced and identified was very much North American lineage. But we know that, especially in Chile, it's a really interesting pattern in that when you're in northern Chile and you look at viruses coming out of wild birds, they're all North American lineage. When you get to around Santiago, that middle area, you start seeing primarily reassortments between North American and South American lineage. And then southern Chile is almost all South American, like they're coming out of Argentina. So we would expect, or we're expecting that depending on where those H5s are being isolated from, we may see very, very different types of viruses.
Thank you very much, Stacey.
Okay, and the next question is from Dr. Todd Hatchett. How do you tease out whether the lung nasal samples with the higher CTs in the contact animals represent environmental influenza and not infection? I presume for Dr. Warner. And I would encourage others, if you want to post questions, to continue to do so. Over to you, Bryce.
Thanks. Yeah, I mean, it's a good question. I think for that particular experiment, I mean, we did have positive samples that were swabs and tissues, and so I think I think that was more of a concern for the swabs, not knowing sort of, you know, if we're swabbing the outside of the animal, maybe that's just sort of environmental contamination, versus, you know, I think it's safe to say that, you know, if you see it in the lung, that it's, you know, likely not, that it's, you know, the virus is down deep in the lung. So that, you know, I think that's not as much of a concern. We have done environmental swabbing of the cages in these studies and seen, you know, we do get PCR positive hits when we look for environmental contamination. So that is a concern, but I think, you know, focusing on the tissues in this case sort of, you know, reduces the chances of that being an issue.
Thank you so much, Bryce. And if there are no other questions, I do have one for Dr. Miller. I'll go ahead and ask it. Matt, you know, we've heard about how the viruses are reassorting quite successfully, and we're seeing combinations of both Eurasian and North American segments. Presumably, the HA seems to be relatively stable or consistent, but in this kind of shifting landscape, how do you even begin deciding which are the best candidates? I mean, now we're in a position where we almost have too many, and it's a very different position than what we were in maybe 2 years ago. So how do we even begin rationalizing? What's the approach?
Yeah, it's a good question. I mean, I think one of the biggest limitations right now is that we're still stuck in this paradigm of disproportionate focus on the hemagglutinin, and we know that is a bit of a dangerous bet to hedge on. I think that one of the ways that it's likely that we can mitigate disease in the context of a pandemic is to generate vaccines that also create strong T-cell responses against conserved internal proteins. So, you know, the polymerase, nucleoprotein, those proteins tend to be very, very highly conserved, even in the context of the diverse influenzas that exist in avian reservoirs. And there's quite a bit known now about sort of epitope mapping of the T-cell epitopes and their mapping onto different human HLAs. So I would like to see us, you know, collectively kind of move away from vaccines that focus only on hemagglutinin for that very reason. I think it leaves us in a position where, based on the current regulatory frameworks, you know, there are these sort of exemplary candidate vaccines that get approved based on immunogenicity studies, because we have a correlative protection for flu in the HPAI assay. But it's a dangerous game to play, because it puts sort of all of the eggs in a single basket, so to speak, no pun intended, with egg-based vaccines.
And actually, go ahead, Jimmy.
Yeah, just one last question for Dr. Miller here. For the SARS-CoV-2 vaccinations in mice, where you didn't see sterilizing immunity or reduction in virus titers in the lung, but there was a reduction in pathogenicity, what were the clinical readouts, Matt?
Yeah, that's a great question. So we do sort of gross readouts in monitoring the animal's weight, as well as in other clinical signs, you know, hunching, grooming, all of those sort of typical clinical signs that are monitored in mice. We also do really detailed histopathological examination of lungs, though. And what I didn't have time to really go into is that what we see in the context of trained innate immunity are lungs that look almost like uninfected lungs. So if you don't have intact trained innate immunity, you can see just on gross examination, sort of hemorrhaging and destruction of lung tissue. When you do histological examination, you see lots of the classical immune cell infiltrates, destruction of airways. When trained innate immunity, though, is intact, there is almost no tissue destruction at all. It's nearly impossible to distinguish an infected animal from an uninfected animal, despite the fact that those infected animals have viral titers that look like infected unvaccinated animals. So it's really quite striking.
If there are no other questions, I will take the liberty of asking another one very briefly. For Dr. Schultz-Cherry. Stacey, this is a question that I hope also makes Damien smile, because it's something that we've been thinking about a little bit. I think we're all familiar with IRAT and TIPRA and these tools to do risk assessment that are very focused on human health. But as we're now seeing all this spillover into mammalian populations, and I'm sure people have seen the reports, for example, about polar bear, and we're seeing it in threatened and also other species, but yet we have virtually no tools or really even the infrastructure, nothing at least compared to what we've been doing for human health to really do risk assessment for species other than humans, and especially wildlife species. So I don't know if you're aware of any movement, if we really are going to do this as a One Health thing, we shouldn't just be looking at the risk to humans, right? So I guess I'm just looking for your thoughts, and if you've had any of those discussions with your colleagues.
Yeah, it's a great question, and we have had those discussions with our colleagues. Our risk assessment pipeline, there's that human health component, and then we look within different populations. So a lot of it is, okay, is this going to be a problem for the poultry industry? But we are thinking about ways to expand that into other species. So one example, and it's not ideal, but John Runstadler's group at Tufts University has developed murine mammal, I think they're seal or sea lion primary respiratory cells. And we're doing the same thing for a number of different species, and we're going to work with people in Cambodia to get into some of those really sort of exotic wildlife. And you make nasal, you make bronchial, and you make alveolar, you can put them back together. We're doing a lot of work to make more complex, almost like a lung and a chip. And while it's not an ideal situation, it's clearly not infecting the polar bear. If we can make enough of these cells, it'll at least give us some kind of idea of, can these viruses even infect them? And that's something we're going to look at when with our Chilean viruses, is just ask ex vivo in primary swine respiratory cells, can we get an infection? And I think that's one way to start, but we need to reach out and work more with our wildlife colleagues and figure out the best way to investigate and really identify who are these at-risk species. Even our companion animal people, we have a lot of vets in South America saying, do we need to be looking at dogs and cats and horses? And we're like, absolutely, because we don't know.
Thank you.
Thank you so much, everyone, for those questions. I would like just to end the session by thanking all the speakers once again for these fantastic talks. Just some highlights for me. Damien told us about the omnipresence of HPAI across this Canadian country. Fantastic work. Stacey, of course, with her groundbreaking field work in South America. Bryce's expansive H5N1 in vivo characterization. And of course, Matthew's pioneering methods to generate even better vaccines and those thought-provoking questions at the end. A really great panel and great talks. And thanks, of course, to the attendees. And most importantly, of course, thanks to my co-host, Samira Mubareka. And again, the fantastic funding provided by CHR but the masterful organization of CAR, the Canadian Association for HIV Research, Aaron Love and Andrew Macejic. Sorry, Andrew, if I mispronounced your name, I always try. But thank you, everyone, for this great session. It's truly been a pleasure from Sam and myself. Thank you.